Okay, so we're going to talk about we're going to talk about innate immunity. Um, and then the next section is going to be adaptive or acquired immunity. And then we're going to kind of pull those together to talk about some hypersensitivities. So uh, our time spent on innate immunity really should just kind of be reviewed. Now, we're also going to talk about the inflammatory response, which is a pretty classic part of pathophysiology, so we'll spend some time with that, and we'll spend some time with some other stuff, a lot of things actually. So hopefully we can kind of keep this short, but I don't know. So immunity. What is immunity? It works in, I guess it depends on the book or what you read, but several different stages. Your first line of defense is what we're going to kind of focus on, which is innate, also sometimes called natural or native immunity. I don't really like any of these terms because really the most important part of this is it's non-specific. So when you get injured, if you get a splinter or something like that, well, there's a whole bunch of stuff that goes in there. Yes, there's bacteria, but there's also little pieces of wood shavings or whatever, if it was wood. And so all of those things just need to be, you need to just get rid of that stuff. You need to either break it down or something, or eject it back out. But that's really not specific. So even though there are bacteria that are found in there, we have an immune response that will just see that as being foreign. It'll say, I don't know what that is, but it's not really from this body, so we're just going to get rid of it. That can also be like our own cells. If our own cells are kind of not really producing the right proteins, and your immune system doesn't recognize it, it'll take that out. That's not specific. Now we get into I'm going to kind of jump down here, but the third line of defense, which is specific, then it's going to say, okay, here's a virus here, and I recognize that virus specifically as being bad, and so I'm going to kill that virus or this bacteria. But the specificity, it goes beyond that. It's not just that it sees that virus and it ignores the other virus, but what happens with adaptive immunity or acquired immunity is that it will launch an antibody response to it, and so it's got all kinds of stuff in place to specifically hit that virus hard. Now, there may be another virus over here, and these guys just won't even notice that. They're going to notice this one. Now, you will build up adaptive immunity to this one as well. So that's the big difference is there some kind of specificity going on? And in that first line of defense, that innate immunity, there isn't really specificity. Not really. We'll talk about how it can recognize that it's a bacteria versus a human cell, but that's really it. So the first line of defense is innate. Second line of defense is inflammation, which kind of works along with this innate immunity. And then the third line of defense is adaptive, and we'll have a whole other section for that. So innate versus adaptive, I kind of went through this already, but we have to clean up after pathogens as well as dead cells, foreign debris, all of this junk that kind of gets in there, and that's what the innate immune system is primarily for. So there are two basic responses to do it. There's innate and adaptive. So adaptive is going to be more focused on pathogens and sometimes allergies. But innate is nonspecific. It will attack anything that's non-self as well as our own dead and defunct tissues. It's faster. It's not selective, and that's kind of important. It's faster, but that has some caveats. The second time you're infected with like a cold or flu or something like that, the first time your innate or your adaptive is very slow, but the second time it's actually the fastest. Okay, so but in general, for a new type of infection, your innate immune system is going to be right on top of it. Okay, so it's faster, it's not selective, comprises any white blood cell except what we would call lymphocytes. Okay, and lymphocytes are T cells, so T helper cells and cytotoxic T cells. And also, do I have that in there? And also B cells. Okay. Now note what I'm saying here is that it's all white blood cells except the B cells and T cells. All other cells are part of the innate immune system. Remember that. So that means macrophages, dendritic cells, the eosinophils, neutrophils, those are all part of the innate immune system. The T cells and B cells, which I have down here, are part of the adaptive immune immune cell or immune system. Okay, so it does not distinguish between microbes. It just attacks them all with the same intensity, and it's also part of this inflammatory response, the innate immune system is, or the innate immune response. So adaptive immunity, also called acquired, and I use those terms kind of interchangeably. So adaptive, acquired, it's the same thing. It's specific, which means it recognizes and kills specific pathogens. Okay. And we'll see how that's important later on. So it's a stronger, more prolonged response to that specific pathogen. Humoral, and again, we'll talk about this in detail, but humoral, just humors means fluid, so it's floating around, so that it can kill things that are floating around. It can kill things that are hiding in cells. So that's actually something that the innate immune system is not able to do. It's not able to kill something. If something hides in a cell, well, it's not gonna find that unless there's something wrong with the cell, and then it'll just kill the whole cell. Okay, so part of the innate immune response, barriers. So we have barriers that try to keep them out to begin with, so we don't get infected. So skin is a physical barrier. It has a bit of a lower pH, 5.5, and there are also friendly bacteria that can block pathogenic bacteria. We see this in the intestine. A lot of times there are potentially pathogenic bacteria, but we have so many other just harmless, sometimes helpful, other types of bacteria that these pathogenic bacteria never get a chance. So that's the biggest key, is to try to keep them out. Stomach acid helps do that. The pH is very, very low. It's basically just hydrochloric acid in the stomach, and that will just kill bacteria on contact. So if you eat something that has bacteria on it, then it will kill most of the bacteria. Now there are some bacteria that can get through, and we know that because we have gut bacteria, and we can get certain illnesses just because the bacteria can't get through. But for the most part, that's what the stomach acid is doing. It's also breaking down denaturing proteins, but so it does that too. But we're talking about immunity. So mucus and cilia. So the mucus so we have cilia, which are these little things that kind of move. The cilia are these things that create that wave movement. So they can sort of move mucus along. So that's part of it. Mucus is that thick, mucus snot kind of stuff, and that can also trap pathogens. Earwax, kind of the same thing. It's got certain chemical makeup. It's sticky. Stuff gets trapped in it, and they die. Tears have antimicrobial properties, and they kind of keep flushing stuff out. So all of these things are barriers to prevent an infection in the first place. But pathogens can still get through. So we have injuries can allow them access. Some bacteria make it through the stomach. Some bacteria or microbes may compete with natural gut microflora to invade. And I say microbes because that can also be like fungal. We have fungal infections, we have viral infections, we have all those kinds of things that can happen. So pathogenic bacteria have evolved ways to take advantage of weaknesses, pathogenic bacteria and viruses. Okay. Fungus have a little bit of a harder time, and that's usually just when people are immunocompromised. But these bacteria and viruses have evolved ways to take advantage of weaknesses or to overcome these barriers. Okay, so innate versus adaptive continued. We can see over here, so we're going to get into a little more detail. We can see over here there's a microbe, and that microbe moves in, and we have phagocytes. So this is primarily, we're thinking of things like macrophages that will just recognize this as saying, nope, you're not part of this body, and it will eat it. We have things called natural killer cells, and natural killer cells can also recognize like cells that aren't producing something called MHC and tumors and things like that, and say, okay, that's not right, and can kill those. These are all part of the innate immune system. We also have something called complement, and that's a whole bunch of proteins that are floating around in your blood right now that can form something called a membrane attack complex and can poke holes. Essentially, that's what they do. They poke holes in bacteria, and they can also affect our own cells. but usually it's not going to be a functional cell. But they can poke holes in it, and then that will cause them to die. The adaptive immune system, I won't say too much about it since we're covering it later, but that's where you have like antibodies, your T cells, and they're very specific. They have very specific receptors. This TCR stands for T cell receptor, but they have very specific receptors that bind specifically with a particular microbe. Okay, so key cells. So of the innate, the phagocytic white blood cells, neutrophils, macrophages, dendrites. I guess these aren't all phagocytic, so it's kind of poorly labeled. But these are cells of the innate immune system. So neutrophils, macrophages, dendrites, mast cells, natural killer cells. So these are, and you're gonna have to learn a lot of this, and we'll go over specifically what they do in the next couple of chapters. So you will know what these are, but and we'll cover them here too. And then the adaptive are the B cells and T cells. Now we do have something called antigen presenting cells, which is where the innate immune system, so you see up here macrophages and dendritic cells are listed as innate immune cells, but they will carry that signal, so they'll engulf something, and then they'll carry it to the B cells and T cells, and they'll say, hey, what is this? Do you recognize it? And then the B cells and T cells are going to be able to launch a specific defense against that particular thing. So sometimes these guys work together. So even though macrophages and dendritic cells are part of the innate immune system, they do work with what we call the lymphocytes because they're found in lymph primarily, these B cells and T cells. The macrophages and dendritic cells will work with B cells and T cells to form an overall assault on anything that's attacking you. So let's break this down more specifically. So granulocytes are neutrophils, eosinophils, mast cells, basophils. Okay, so what we mean by a granulocyte is it's really pretty simple. They have granules, and you can kind of see these in these pictures. You can see the little dark spots. These are granules, and these granules have a lot of different things. Primarily what we think of is histamine, but they have several different molecules. In the case of eosinophils kill like multicellular even organisms, so worms and things like that. And these have toxins, so they have toxins that are released when they're activated, and they can kill anything surrounding it. Unfortunately, they also kill your own cells. So an immune response, while it's helpful, you don't want constant activation of the immune response because that will actually weaken your body in some ways because it can kill other cells in the area. So neutrophils, eosinophils, mast cells, which are related to basophils. It's really there are a few differences, genetic differences, but mainly it's where they're found. Neutrophils, also referred to as polymorphonuclear neutrophils, and I say that because a lot of times on a complete blood count you'll see PMN instead of neutrophil. Okay, so you have to recognize at least this three-letter term, and that's really kind of descriptive because if we go up here and we look at the neutrophil, we can see that the nucleus is kind of crazy. Okay, it looks like it's sectioned into three chunks. Same with the eosinophils, but that's why it's called polymorphonuclear, because this nucleus is kind of separated into little chunks. It's still a nucleus, but it's just not this nice round looking thing like we're used to seeing when we think of a nucleus. So the neutrophils, okay, so let's focus on neutrophils. They are the first responders. So that splinter I was talking about earlier, the first cells that are probably going to find it, there may be some mast cells sitting in there, but the first cells really that find this stuff are neutrophils. And they come in fast, and a whole bunch of them, they live fast, die young. But they predominate in that early inflammatory response. So when you first get poked with something, these neutrophils will move in and launch an early response. They ingest bacteria, dead cells, cell debris, so they start really kind of clearing stuff up. They don't live very long, which is good because you don't want them to be active for too long of a time. You want them to kind of clear out their stuff and then go away so they don't start damaging friendly cells. But they are associated with pus. So when you think of an injury that you've ever gotten that produces pus, those are neutrophils. Those are dead neutrophils. Okay, so they're the first responders. They don't live very long. They produce pus, which we will call a purulent exudate. So an exudate is just something that is kind of moved out into the extracellular fluid. It exudes out, and purulent refers to pus, and that's what the neutrophils really are, the pus. I mean, it's all those cells that are dead that make up the pus. So let's talk about monocytes and macrophages. Now, these are the same thing, really. Okay. Now, I don't want to be too cavalier in saying that they're the same thing, because they change. So monocytes are what are floating around in this is the capillary. But then when that monocyte moves out into the extracellular fluid, it changes into a macrophage. Okay? So they're the same basic cell, but when they're floating around, they're monocytes. And then when they move into the extracellular fluid in the interstitial space, they become macrophages. And you can see that they're morphologically different, which means that they look different because they've been activated. So the monocytes that are activated become macrophages, and that happens when they move into the tissue. So monocytes are produced in the bone marrow, okay, and really all of these cells are produced in the bone marrow, all leukocytes. We're going to see that word, so I'll just mention it now. Leukocyte. Don't get that confused with lymphocyte, but leuko means white, so these are white blood cells. They were named because they appeared white as opposed to red blood cells. Okay, so they called them leukocytes. But all blood cells, including red blood cells, white blood cells, platelets, they're all made in the bone marrow. Some of them will mature in the bone marrow. Some of them, like the lymphocytes, the T cells, will mature in the thymus. That's why they're called T cells. But anyway, so they're produced in the bone marrow, circulate in the blood, and migrate to the inflammatory site where they develop into macrophages. So macrophages are mature, activated monocytes that are found in almost all tissues. They typically arrive later. Remember, our first responders are the neutrophils, and the neutrophils will lay down a signal saying, hey, there's something going on here. We need, some big guns. And the macrophages are these larger cells that are good at engulfing or phagocytosing stuff. Okay, so they arrive at the inflammatory site about 24 hours or later after the neutrophils are there. Their functions, phagocytosis, they will release signaling molecules that will induce and continue in the inflammatory response. So they signal proteins that activate other immune cells. They really just activate other immune cells. And they're general scavengers. They clear out dead cells, cell debris. They can, that's kind of their job. They're these just big cells that are kind of engulfing anything that they see as foreign. Now, they also act as antigen-presenting cells. We're not going to get into too much of what that means, but that means they're going to send that signal up to the B cells and T cells, those lymphocytes. I know. A lot of weird words. So dendritic cells, they get their names because they look like they have dendrites. They have nothing to do with neurons. They're not neurons. They're not associated with neurons. Completely different cell type. They just happen to have these little pokey things sticking out. Somebody says, hey, those look like dendrites. So let's confuse future science students by calling these dendritic cells, even though they have nothing to do with neurons. Okay, so they get their name because they look like they have dendrites. They're not related to neuronal tissue. They are definitely leukocytes, or white blood cells. So they're typically found under epithelial tissue, which is skin, and skin that's also found all over your organs, lining your organs. But that's where these dendritic cells are mostly found, but they can be all over. Capture foreign agents, transport them to regional lymph nodes. So that means they work as what we call antigen-presenting cells. Okay? So that's an important role for these guys. So they mature in the lymph node, and take on antigen-presenting roles, which we'll cover when we talk about adaptive or acquired immune system response. Release molecules that help direct the adaptive immune system. Now all of these, and really we have a general term for this, but cytokines are communicating molecules that are very prominent in the immune response. So it can release signaling molecules, and then those signaling molecules can activate other parts of the immune system. So how do cells of the innate immune system recognize invaders without being selective? So how do they know? How do they know this is a bacteria and not one of my cells or your cells? How do they know that it's one of your cells and not my cells? So in general, in the basic thing that it can do is there are certain things, certain molecular patterns that are different. It's kind of like you don't have to know the identity of every person walking around, but you can definitely tell that a dog is a dog because it's covered with fur. And so it's kind of that same concept. And there are certain patterns, certain molecules that are only produced by bacteria, for instance, and single-stranded DNA is only found in viruses. So we know that there are major differences between these cells, and that's what it recognizes, and it calls them PAMPs, pathogen-associated molecular patterns. So these are conserved patterns on foreign molecules. So, for instance, lipopolysaccharides, those aren't made by mammalian cells. Peptidoglycans, bacterial lipoproteins. Flagella, we don't have flagella except sperm, and sperm is separated from our immune system for that reason, because our immune system would probably flag it as foreign and would destroy the sperm. So it's a privileged it's in a privileged area, the testes, and your immune response can't affect that. So foreign DNA, double-stranded RNA, single-stranded RNA, those kinds of things are also patterns that we wouldn't see in a normal cell, in a normal human cell. So receptors on immune cells, the receptors that they have to recognize these PAMPs, these weird things about other cells, foreign cells. So they have receptors. These receptors are just called toll-like receptors. It's based on a fly protein where they were first discovered. But anyway, those are what recognize these pathogen-associated molecular patterns. Okay. So inflammation, let's talk about inflammation. It's an innate automatic response to cell injury that neutralizes harmful agents like bacteria, removes dead and damaged tissue, generates new tissue. Okay, so it starts that proliferative phase where it starts to generate new tissue, promotes healing, so it can activate certain growth factors to cause cell division that can cause, if there's an injury, that to be repaired. And the whole idea is to minimize the effects of injury or infection, to get on it early and try to take care of it to get you back on the healing road as soon as possible. So inflammation is necessary, but if it's prolonged, it can lead to unwanted effects because these are releasing chemicals. They're trying to kill other cells. So a lot of times that can come back and actually do more harm than good in some cases. So inflammation in general, damaged cells release inflammatory mediators, and then so that's the damaged cells. When something breaks, if a cell breaks, there's stuff that's supposed to be inside the cell that's now being released outside the cell, and that's an indication that there's a problem. So there's a local response, so we'll talk about these in more detail, but there's a vascular stage, and then there's a cellular stage. So that's a local response. Then there can be a systemic response. Usually we know that there's a systemic response. The main reason is we'll start to develop a fever, which we'll talk about later. But a systemic, it's the whole body response, and then you've got other blood cells being produced, so you might have an increase in neutrophil production. So that's when you get the complete blood count. You look at, you test the blood and you say, oh wow, your white blood cell count is high. That's kind of an indication that your bone marrow is making more white blood cells. That indicates that it's not just local anymore, that now it's systemic. Okay, so white blood cell response, and then there's what we would call an acute phase response that happens a little more quickly. All right, so So let's look at these granulocytes. So white blood cells, or leukocytes, in inflammation, there are two categories. There are really granulocytes and agranulocytes. So let's first talk about the granulocytes. So we mentioned the neutrophils as being the first responder. They're the most plentiful. 60 to 70% of the immune systems in your body right now are neutrophils. They're short-lived and they are the origin of pus. Eosinophils are associated with parasites. Okay, so worms, that's what I think of. I think eosinophils, I think worms, right? Involved in the allergic response, so this also has an allergic reaction. And in fact, I have this little note over here. Eosinophils, mast cells, and basophils are all involved in the pathogenesis of allergic disease. So all of these guys can be increased in as part of an allergic response. Okay. And can trigger an allergic response. And we'll talk about hypersensitivities later. So associated with parasites involved in allergic response, mast cells localized in tissues, and these are really specific. I couldn't decide if I want to say specified or specific, but they're specific for for allergic responses. So pollen is, kind of an accidental. That's a hypersensitivity. Maybe we don't want to be allergic to. We don't want to have an immune response to pollen, but that's going to that can happen. But they do serve a good purpose to try to very quickly neutralize an invader. Basophils, these are associated with the amplification of an allergic response. So these are all allergic. These are all also associated with the allergic response. And we'll talk a lot about mast cells later on when we talk about hypersensitivities. So depending on the cell, the granules contain a range of chemicals. So I already mentioned histamine. They also may have heparin, which prevents clotting. Toxins to kill invaders, especially your eosinophils, can be very toxic. Proteases, which break down proteins, enzymes to synthesize prostaglandins and leukotrienes. We haven't talked about prostaglandins and leukotrienes specifically yet, but these are cell-mediated inflammatory molecules. Okay, prostaglandins, that's what you block when you take an aspirin or an NSAID. You block the production of prostaglandins, and that can we'll talk about how that can reduce inflammation. Okay, but anyway, these guys are all in there, and they all have signaling molecules, cytokines, chemokines. We haven't talked about what chemokines are yet, but we will. Okay, and then there are the agranulocytes. So macrophages are agranulocytes. So they don't produce granules. Their big, powerful superpower, I guess, is phagocytosis. So inactive monocytes circulate until they receive a chemotaxic signal. All right. So I'll go ahead and mention what a chemotaxic signal is. If we have a blood vessel here, and here's a monocyte that's floating through here, there are signals being produced by neutrophils, or it could be produced by the organism that's being attacked in general. So, but there are little breadcrumbs, little chemicals that that are being produced out here. And this will direct that macrophage right to the area of where this infective or this inflammatory process is taking place. So they will follow that chemotaxic signal. Taxis means to move, chemo means chemical, so it's a chemical signal where they are taxiing. Okay. Then as they move into the interstitial spaces. Macrophages, I'll show a picture of that in a little bit. Macrophages phagocytose or eat and digest invaders rather than secreting toxins. They also produce their own signaling molecules to prolong that immune response. Okay. So inflammation. Normally, if you have no injury or inflammation, then you have this nice, everything is in its place, everything's where it's supposed to be when everything's working normally. You have a selective barrier for you in your blood vessels, and so that keeps most microbes out and controls what can move in and out of vessels. So it's only when those things start to cross that we know that there's a problem. So the endothelial cells lining the vessels produce agents that keep them open and unobstructed. We do have vasoconstriction, vasodilation that can take place, but really we don't want any clotting to take place. So there are signaling molecules saying don't clot. nothing's wrong, everything is fine, and these guys will just keep moving. The platelets, basophils, the eosinophils, lymphocytes, they just kind of keep moving as long as there's really no signal. Blood vessels are non-thrombogenic, which means they don't produce clotting. Platelets are unable to bind to other platelets and cells, so clotting is not supposed to occur when everything is happening normally. Now, there are some molecules that are outside that aren't supposed to be in the blood, like elastin, and certain other fibrous, this is a fibroblast, which means it's going to produce other fibers like elastin. But collagen is a big one. So collagen is supposed to not be in the blood vessel. It's supposed to be outside the blood vessel. So we know that when we have a break, and now we have collagen, so we have exposure of the blood plasma to collagen, then this is how we know. This is one of the signaling molecules. There's also like tissue factor and stuff like that. But that's how we know that there's been a break or a breach. And that can then trigger all of these immune cells to move in. And of course, neutrophils are going to be the first ones. Okay. Then we do have macrophages and mast cells that will move out, and they do exist out in the tissue. So many cells and other chemical factors are circulating, but do not cause inflammation unless they get a signal to do that. So let's say that we do have an inflammatory response. So the early or immediate local reaction to injury. So that's what our acute immune response is, the early or immediate local reaction to injury. Something happened, something broke, something went wrong. And so we triggered this acute inflammatory response. And the whole idea of it is, like we said earlier, designed to remove the injurious agent and limit tissue damage. It's triggered by infection. So if bacteria get in there, immune reactions that can also trigger inflammation, which we'll see about kind of when we talk about hypersensitivities, especially type 3. Hypersensitivities, physical or chemical trauma, something is broken. So there are cardinal signs of inflammation. So once this acute inflammatory response takes place, then we see the same types of things, and that's rumor, tumor, calor, dolor, functialesa. So that means redness. Okay, well that's going to be because you have increased blood flow to that area. Okay, red blood cells are red, and so you're going to see redness. By the same token, you're also going to have heat because of that increased blood flow moving in there. You're going to have remember we talked about edema. You're going to have more cell capillary permeability, so you're going to have a buildup of fluid, which is going to cause tumor or swelling. And then pain. There are pain signaling molecules that when things break open, it releases certain chemicals that will activate neurons to cause a pain response, to say, hey, something's happening here, and it gives your brain some information saying, hey, you might not want to walk on that foot for a little while, and so it will send that as a pain response. And then functialesa, or loss of function, just means that things aren't working right, and we have some broken cells. Things are not doing what they're supposed to be doing. Okay, so stages of acute inflammation. Vascular, fluid moves to tissue. So one of the immediate things that happens is you have immediately vasoconstriction. So if you've ever tested yourself for like a blood glucose test where you prick your finger, you notice that immediately there's no blood, and that's because you initially have vasoconstriction, but then you get vasodilation, and with that injury, we start to allow fluid to move into the tissues. Well, one of the reasons for having fluid move into the tissues is to start to get nutrients in there, or ultimately to allow cells to move into the tissue. And that's where we kind of back up and we say, okay, well, that's the cellular phase or stage, because now we're letting cells move in. These aren't very good pictures, but that's just showing cells kind of moving in, and I have these pictures a little bit better in another slide. So let's first talk about vascular. So there's a quick period of vasoconstriction followed by vasodilation. Okay, and what's the vasodilation going to cause? It's going to cause redness, okay, rubor, and calor, which is heat. Okay. So it's going to be warm and it's going to be red. Okay, so increased blood flow So a quick period of vasoconstriction followed by vasodilation. So I have increased blood flow, but it's slower. You initially have that vasoconstriction, and that's going to cause that redness and heat. So increased blood flow leads to net flow of fluid and plasma proteins out of vessels. So no, we're not talking really about cells yet. We're talking about fluid moving out, proteins moving out. Out into the tissue, out of the blood vessels into the tissue. So that's going to create swelling. It's also going to trigger pain. We have certain, like substance P, we have certain chemicals, bradykinin, but we have certain chemicals that will directly bind to a receptor and say, okay, send a message to the brain that something's broken here, and that message is going to be, like I said, pain. And then it's going to impair the function. Okay, so all of these cardinal signs. So the cellular, and then we move to the cellular stage. So now damaged tissues and resident immune cells like macrophages and neutrophils will release chemical signals indicating damage has occurred, and then the gaps form, further increasing vessel permeability. So remember, we have these endothelial cells lining our capillaries. Well, these will constrict and leave pores, and these pores allow macrophages and neutrophils and other immune cells to move out of the vessels and into the area where the damage has taken place. Okay? So circulating neutrophils are attached to the site by adhesion molecules. So normally these neutrophils are just kind of rolling through, rolling, just fine. And then when a signal happens, what happens is there are these little binding proteins that will, or adhesion proteins, that will grab those neutrophils and say, whoa, stop. And then that neutrophil will stop and it will transmigrate, diapedesis, and move into these areas. Same thing with macrophages. Macrophages will do this too. And it will move toward where the bacteria are. So chemotaxis, chemokines, are signal molecules that are released from the damaged area, and the neutrophils will migrate towards those signals and phagocytose the invaders. The neutrophils will then produce its own chemokines, so these signaling molecules, which will then direct macrophages to that area as well. So neutrophils migrate towards these signals and phagocytose the invaders. Eventually more macrophages are attracted that also phagocytose invaders and damage cells. So this is what's happening. This is our, a couple of stages of the inflammatory response. So macrophages may activate lymphocytes, causing a longer lasting response, and that's where it triggers the adaptive immune response. It really depends on whether or not your body can manage it locally before it goes systemic. It tries to manage it locally. So this is just another picture of the same thing. You can see these neutrophils rolling along, and then those adhesion molecules say, whoa, we've had some damage, and we'll stop them. You've got increased permeability, so the neutrophils can move through. They move through, they start to engulf things. They send out more, so they'll activate the macrophages. In this case, you see the macrophage is producing these chemokine signals. It's laying these breadcrumbs down saying, hey, damage is here, and that's going to then cause other immune cells to also move in, and just more and more. It's kind of a positive feedback kind of a thing. So it's a snowball effect. Okay. So whatever, but something has to initially give that signal. Usually that's some kind of tissue damage, and then everything else will kind of move in to try to take care of it locally if it can. So inflammatory mediators, what chemicals are responsible? So this is a graph I have because we kind of have to know the difference between cell-mediated and plasma-mediated, or cell-derived, plasma-derived. So if it's plasma-derived, when you see the term plasma-derived, do yourself a favor and start thinking, okay, the liver is making molecule or making chemicals somewhere. Well, it's making it in the liver. But the liver is making chemicals and then releasing those into the plasma. It's usually the liver that's doing it. So these are plasma-derived mediators, some things that it makes, acute phase proteins, pyrogens, which that can be found factor 7, so that's always floating around. It's a clotting factor. Complement proteins made primarily by the liver, and those are also floating around. So these things are just in your plasma, so we call them plasma-derived. Now, there are certain cells, so mast cells, platelets, neutrophils, leukocytes, all of these things that we've already mentioned, macrophages, lymphocytes, all of these guys can also produce their own chemicals, okay? And so those are, we call those cell-derived because they weren't floating around in the plasma to begin with. They were made by the cells. And we'll talk about, specifically, we're going to talk about prostaglandins, but there are others: nitric oxide, lysosomal enzymes, reactive oxygen species, which can damage things, histamine, serotonin, which are involved in vasodilation, vasoconstriction. Okay, so plasma-derived mediators, proteins produced in the liver and released into circulation. These molecules are not reactive until activated at the site of inflammation. So kinin's the best example is bradykinin. Bradykinin causes vasodilation and pain. Okay, so when it is activated, as it's floating around, it's not doing anything; it's minding its own business. But then, when there is some kind of damage, other molecules are released. The bradykinin becomes activated, and it will trigger vasodilation. It will trigger pain, and we'll talk more about bradykinin when we talk about clotting, because it's also involved in that. But it also sends a signal when it's activated. It sends one of these pain signals, and it activates receptors that will then trigger that pain response. Coagulation and fibrinolysis proteins. So fibrinogen and fibrin are involved in the clotting process, but they're kind of going around. Fibrinogen, we'll talk about it again when we talk about clotting, but is doing nothing. I mean, it's floating around in your blood right now, but if it's not activated, then it doesn't do anything. But when it's activated, it can cause clotting. Okay. Fibrin is a mesh, which is going to cause that, which is going to actually cause the clot. It's going to cause those platelets and all that stuff to kind of stick together. Okay, and complement. Complement is that there are also a bunch of circulating molecules that come together to create pores. So here's a picture of a pore or membrane. The membrane attack complex is really all the proteins coming together, but ultimately they produce pores. They poke holes in any kind of bacteria, and that's going to kill the cell. But anyway, the important thing to remember here is if it's plasma derived, that means they're floating around right now. Okay, so that means they're in your plasma right now. Cell derived are released when the injury takes place. So cell-derived inflammatory mediators made in cells and released when the injury occurs. Histamine and serotonin, so that's part of these granulocytes. It's part of the granules that we see. So inflammatory triggers released by granulocytes, and in the case of serotonin, serotonin is released by platelets, but that's cause dilation of arterioles, increased permeability of venules. So that's a couple of the roles that they have. But the important thing right now is to remember that these guys are produced by cells at the time of injury. Prostaglandins. Prostaglandins are inflammatory molecules that are associated with pain, inflammation, and ultimately can also lead to fever. Okay, so omega-3 unsaturated fats. I'm not going to say too much about that, except that they can form different degrees of inflammation. So a lot of times we talk about fish oil and how fish oil reduces inflammation. Well, it just makes a different subset of prostaglandins. So cytokines, chemokines, so these are inflammatory signaling molecules. So you'll see a lot TNF-alpha, interleukin-1, IL-1, and what these do, and the reason these are important, and the reason that you're going to keep seeing these guys mentioned is because these are released by cells during inflammation, and actually they can be released by other cells, you know, like adipose cells sometimes too. But these are inflammatory signaling molecules, and what they do for us is that we can see that if the levels of TNF-alpha are increased, then we know there's an inflammatory process going on. So this is information for us. Of course, they're serving a purpose, but when we test blood and we see, you know, TNF-alpha levels are increased, then we know that there's an inflammatory process going on. So it's good information. Nitric oxide is a vasodilator, and then reactive oxygen species. Those are highly reactive and kind of deadly, and why we don't want inflammation to occur for too long. So let's talk about this right now. I think that understanding this particular pathway is important. Now, let's kind of break it down and try to make sense out of it. So we remember one thing we know from biology, physiology, is we know that we have a cell membrane, and we know that that cell membrane is made up of phospholipids. Okay? So our question here that we're trying to answer is, okay, first of all, what's a prostaglandin and where do they come from? We know that they're cell-derived because we just said that. Well, what happens is that to make a prostaglandin, which is an inflammatory mediator, it breaks off one of the tails of this phospholipid, okay? So it breaks it off. And then through a series, and that's phospholipase is what's going to do that. So it breaks it off, and then there's a series of enzymatic reactions, and it's going to twist it around kind of like a hairpin or a paperclip or something like that. And so while this started out as a tail of a phospholipid, now it's been bent around and it turns into something called arachidonic acid. Okay? So arachidonic acid doesn't really do anything, but the next step, this is what's important, is this arachidonic acid that was ultimately just a tail of the, a lipid tail of a phospholipid, can be turned into prostaglandins by something called the cyclooxygenase. It's an enzyme. So cyclooxygenase makes a few other changes. I don't know exactly where, but it changes it chemically and turns it into prostaglandins. It also turns it into prostacyclin, thromboxane, and but that's what cyclooxygenase does. And then these prostaglandins, what they do now, so it started out as this tail, got twisted around, turned into arachidonic acid, and now there's another enzyme, cyclooxygenase, that turns it into a prostaglandin. So now that that's been made, this prostaglandin can induce inflammation, vasodilation, clotting, fever. All of these things are associated with, and then there's another direction it can go to cause pain. So all of these things are just cell-derived because it was made from essentially the cell membrane of the cell. Okay, so why is this important? It's important because aspirin and NSAIDs block this enzyme cyclooxygenase. That means that, yeah, arachidonic acid was made, but it never turned into a prostaglandin. So that's why aspirin is anti-inflammatory, okay? And that's why aspirin, this is important, that's why aspirin can prevent clotting. Okay, so thromboxane is involved in clotting. So thromboxane isn't produced in as high of amounts, and so it can reduce clotting, it reduces fever, it reduces pain, even though I don't have pain listed up here. Okay, so this is why it's important. That's why prostaglandins are important, because they induce all of these things. NSAIDs, so non-steroidal anti-inflammatory drugs, that's what NSAID stands for. But aspirin and NSAIDs block all of these things. They block the production of prostaglandins. So leukotrienes, which are made in a different enzymatic pathway, it's called lipoxygenase pathway, produces leukotrienes from this arachidonic acid. But these work with histamines. They have a similar effect, but they're slower, and they are involved in smooth muscle contraction. When I say smooth muscle contraction in this context, think airways. Okay, so think that leukotrienes, when they're activated, can constrict the airways. Think asthma. Okay, so that means by blocking this pathway, then maybe you won't experience asthma, and they're trying to develop drugs based on that. So yeah, that's important in asthma. Now the omega-3s, all they are is they're just a different identity for this little tail of the phospholipid, and they just the prostaglandins, you still produce the prostaglandins, it's just that they tend to not work as well, so they tend to be less inflammatory. Okay, but they still follow that same pathway. All right, so more cell-derived inflammatory mediators, cytokines, molecules released from one immune cell that cause a response in another. TNF-alpha, interleukin-1, activate many other pathways of inflammation. There are over 30 known cytokines. We don't have to know them all, but be familiar with these. maybe C-reactive peptide, which I think we'll talk about. But chemokines released by cells to initiate chemotaxis. This trips people up on exams, so keep that straight because we've got cytokines, chemokines, chemotaxis. I know a lot of words are kind of similar, but if you break them down, taxi, movement. Okay, so nitric oxide is a vasodilator, so if you see that, just think vasodilation. Just kind of a little, trick here. Nitric oxide is associated with an erection, and that's in a roundabout way. That's how Viagra works. It causes the production of nitric oxide, which causes vasodilation, which allows an erection to take place. But we're not talking about that right now. We're talking about inflammation. But sometimes shocking things help us remember. Prevents clotting. That's another thing that can happen with nitric oxide and reduces inflammation. So reactive oxygen species signify cell damage, can trigger cytokine release, or high concentrations damage surrounding cells. All right, types of exudate and inflammation, site and severity. Exudate, remember, exude, the word exude. So here's a blood vessel and things start to move out of the circulation. That's exuding, so we call that exudate. There are lots of different types of exudate that can be made in inflammation. Site and severity can manifest itself differently. So exudate is the fluid and other substances that move into the extracellular matrix. It exudes from circulation and exudes out into, I keep saying that word because people struggle with it sometimes, into the extracellular matrix. So if it's serous is watery, and there will be exam questions, kind of describing this. Okay, a person has a, clear whatever fluid, like they got a burn and they have, what kind of exudate is this? That's going to be serous. If it's watery, it's clear, it's low protein, blisters. So if you've ever popped a blister, it's very watery. Well, it exuded from somewhere, so it is a type of exudate, and since it's watery, we'll call it serous. Now hemorrhagic, that's just blood. Severe tissue injury with damage to blood vessels. That means blood is just kind of oozing out, and that's hemorrhage, and that's hemorrhagic exudate. Okay, fibrinous, lots of fibrin. So fibrin is, we'll find out, is going to be involved in clotting. Okay, so that means that clotting may be taking place. It's trying to hold stuff together, and so it's going to have a mesh-like consistency. So if we're describing that in a test question, then it will say that it has a mesh-like consistency, or grainy or something like that, and that's fibrinous. Membranous, the thing for exam questions anyway, the thing to remember is membranous. If mucous membranes are mentioned, then chances are we're talking about a membranous type of exudate. So they develop on mucous membranes, necrotic cells, and a fibropurulent exudate. Okay, so if it's just clear, then yeah, it might be serous. But if it's fibropurulent, what does purulent mean? Purulent means pus. Okay, what is pus? Leukocytes, or I'm sorry, neutrophils. Okay, so purulent is another type of exudate. So remember, membranous was associated with mucous membranes. Purulent can happen anywhere. So purulent or suppurative, degraded white blood cells. And again, those are mostly neutrophils. Okay. It's mostly neutrophils. Degraded white blood cells, proteins, and tissue debris indicative of a bacterial infection. So remember that. Okay, so if you see purulent exudate, that means that there is usually some kind of infection. That means that you're producing neutrophils. Neutrophils are coming in at high volume, and they're trying to clear something, and that indicates that there's probably a bacterial infection. So may form an abscess, or fibrous proteins may wall off the area, requiring an incision to drain. Okay, so you can form an abscess, and you may have to drain that abscess. A lot of times your immune system kind of gives up. It'll try to get something cleared up for a long time, and at some point it might say, okay, let's call it a loss. We're going to wall it off, and we're just going to kind of let things happen in there without damaging everything around it. And so that's what an abscess is, and that may have to be drained. Otherwise, it'll just kind of persist for a really long time. Okay, systemic. So earlier I said, okay, we were talking about acute or local. Now we weren't able to control it locally, so we have a systemic response, And really the big indicator that there's a systemic response, or one of the big indicators, is fever, but it can also be increased white blood cell counts, stuff like that. So if localized control is not possible, inflammation becomes body-wide when signals are released into circulation. So the acute phase response, acute proteins, that's early on, occurs within hours to days. Cytokines stimulate the liver to produce more inflammatory proteins. So now we're sending a signal back saying, yeah, nice try with what we already had, but we're not getting it done, so you need to make more. So higher fibrinogen, I know we haven't talked about fibrinogen yet, but that is associated with clotting. Okay, so fibrin are they form the mesh and they trap platelets in there and other red blood cells, but they trap all that stuff, and that's what really causes clotting. Okay, and so higher fibrinogen levels. Okay, so those are that's made by the liver, and that's going to increase sedimentation rate. And what sedimentation rate means, INR, but what that means is that if you set some blood out, simple way to think about it is if you set some blood out, it's going to take a little while, but it's going to clot just naturally. Okay, if you have higher fibrinogen, it's going to clot more quickly. It's going to sediment. It's going to form that sediment more quickly. Okay, so that's what sedimentation is. So a white blood cell response. So you're going to see stimulation of white blood cell production. That means the bone marrow is going to, it's the same message. It's like, okay, we tried to clear this locally. We tried to, keep it all to ourselves, but we can't do it. We need more neutrophils. We need more macrophages, okay? So you'll start to see what's called a left shift, and you'll start seeing more immature neutrophils. So normally neutrophils, normally you have the same amount of young, old, or new and mature, but then when you have a systemic infection, what happens is the white blood cells start making a whole bunch of new ones. So you see a bunch of immature neutrophils, or white blood cells. You see a bunch of immature ones, and so instead of having, a nice distribution, you see a whole bunch of these immature ones. So sometimes people will call that a left shift. If you're graphing it out, you can say, oh, a bunch of immature ones over here on the left. If you're saying these are immature and these are mature on the right, then you see a whole bunch of immature ones. Okay, so macrophages may activate lymphocytes for the adaptive immune response. So now we're getting the adaptive immune response in. Overwhelming release of cytokines, and this, if it goes too far and it moves too fast, it can be associated with severe bacterial infections or sepsis. So sepsis is when you have infection that kind of occurs in the blood, and that hopefully can progress to septic shock, but sepsis is just a bacterial infection. All right, so chronic inflammation lasts 2 weeks or longer. So we went from systemic, now it just won't go away. It's inflammation lasting 2 weeks or longer, and it can still be localized. Chronic inflammation can still be localized, but it's characterized by pus formation and discharge separation, which is the discharge of pus, and incomplete wound healing. So it's trying, can't get it done. It keeps trying. So what's causing it? An unsuccessful acute inflammatory response. Okay, so it's trying to do it, but it can't get it done. Low-grade persistent infections or irritants. So something is unable to be removed. So I mentioned earlier that we'll have cytokines or inflammatory signals continue to be produced. Okay, at low levels, it's just there. Obesity actually is adipocytes, so fat cells, have been shown to release TNF alpha, tumor necrosis factor alpha. Well, we know that that is an inflammatory signal, and so that's kind of like this chronic inflammation. So we constantly have the inflammatory signal turned up. Okay, so can lead to cancer formation from DNA damage if you, because remember your immune response is it's involved in cell division, it's damaging things, and then if those things have to keep replicating, then there can cancer formation can occur. Granulomatous formation. So a granuloma is when macrophages, and we'll talk more about this later on, but when macrophages go in, this is actually what also causes atherosclerosis, but macrophages go in to try to clear something and can't do it, and the macrophages will kind of collect, okay, and form a mass. Okay, so it could form a uh, um, caseous necrotic tissue. Um, so we can call that a granuloma. So it's it can be walled off, and so it's when the macrophages are unable to fully clear an invader, so they form a mass. I already said that. All right. So chronic inflammation results in less localized edema and neutrophil infiltration. Remember, we had edema and neutrophils that were that first response, one of the first things that happens with inflammation. But instead, with chronic inflammation, macrophages and lymphocytes accumulate in that damaged area, and they continue to release the inflammatory mediators, these cytokines. It's asymptomatic at first, but it does cause tissue damage over time, and that tissue damage, of course, can lead to cancer. It increases the formation of neoplasms or cancer tumors. So it may damage DNA, resulting in certain cancers. Cervical cancer, liver cancer, stomach cancer are some examples. Granulomatous inflammation can take place. Macrophages mass together around this foreign body that it's trying to clear. Connective tissue will ultimately form. Connective tissue, smooth muscle tissue can ultimately form and wall it off. And macrophages may collect and form foam cells in arteries, leading to atherosclerosis. Now, we're going to get into detail of atherosclerosis when we talk about the cardiovascular system, but for now, that's what we're talking about. That's an example of a chronic localized inflammatory response. Okay, so let's fix everything. So let's say things are working okay, and we have wound healing. I'm not sure where we came up with these terms, but wound closure, you can kind of envision that if you are cut with a scalpel, that it's going to be a nice clean incision, and so all you have to do is pull that together, and there's going to be very little tissue loss, which means very little scarring, and we call that primary intention. Okay, so the wound can be closed. A clean cut or surgical incision is going to be primary intention, very little tissue loss. Secondary intention means the wound is too extensive to bring the edges together. So if we just kind of carve out a chunk, then you can't just bring the epithelial tissue together where it kind of forms nicely and joins up with the rest, and very little scar. So that means extensive granulation is allowed, and you have this filling in with collagen and other fibrous stuff. Blood cells can move into there, and it takes a lot longer for it to repair. So a longer repair time, it's going to be greater scarring, higher chance of infection because it's more open, okay, for that amount of time, which means for caretakers, that's a greater need for care. You need to make sure the bacteria doesn't get in there. It needs to be cleaned. Burns, ulcers, large surface wounds, these are all secondary intention. So as it's healing, okay, so first of all, we have the inflammatory phase. So we have some phases here. This is broken down into 3 phases. So inflammatory phase, the time of injury, that's hemostasis, and we'll talk about that, but that's essentially clotting. Okay, hemostasis is another word for clotting, or what we recognize as clotting. Vascular and cellular phases of inflammation. So we've already talked about inflammation. So that's the first part. Get the white blood cells in there, kind of try to clear out the invaders. Proliferative phase, so 2 to 3 days after the injury, after the inflammatory response has taken place and is starting to subside, building of new tissue to fill the wound. So fibroblast secretion of collagen. So collagen is very common. It's found just about everywhere. And so what happens with collagen is it's laid down by a blast cell. A blast cell just means that it's making new stuff. So fibroblasts will produce this collagen, and this collagen it just kind of forms wherever it forms. Okay, so if you look at the picture I'm drawing, I initially started drawing it all in a neat little row, but really that's not how it forms. It just kind of lays down, and it's very fibrous and holds things together nicely. So it builds new tissue to fill in that space, but that's going to be scar tissue. That's not functional tissue like it was before. So fibroblast secretion of collagen, angiogenesis of new capillaries. I'm going to have to make my capillaries black. But capillaries can form, that's angiogenesis, to give a blood supply to the area where it's being formed. Okay, so you have blood supply there. Granulation tissue is laid down for epithelial cells to grow on, like a basement membrane kind of thing, but you have to have everything growing in some kind of an organized way. It's going to look a lot different though. But then we have what's called maturation or remodeling. So 3 weeks after the injury, loss and gain of collagen reorganizes the wound for maximum strength. Now, if we think about what that means, if we go back to this, what was originally drawn here, and where the collagen fibers are going every direction. Well, as you use that area, let's say that it happens like on your elbow or something like that. Well, it might be stronger if the collagen is all aligned this way. So remodeling can take place over time as you use it, and it may lay down. And so these guys that are moving sideways may be damaged or eliminated and go away, and only the ones that are really working and serving a function will remain, and that's remodeling. So it's trying to make it the strongest it can be. Okay, so that's maturation or remodeling. Okay, so this is kind of an example. This is a figure that shows these, this shows primary intention and secondary intention, but it kind of takes you through the phases, and you can see that in primary intention you really don't have that much of a scar, but in secondary intention you are going to have more of a scar. If you've damaged right through, you know, the where you can't pull that epithelial tissue back together, then it just kind of fills in with collagen, and the cells that do form there are a little more random. Okay, the last thing we're going to talk about is fever. Okay, so fever, again, that's a big indicator of an inflammatory or a systemic inflammatory response or an illness, and that's really the key. Okay, so we have lots of people who say, with the purpose of a fever, well, you know, it kills bacteria. Well, it might, and it does in some cases, but there's never been anybody we've never really said absolutely this is why we get a fever. We know this. We suspect, and there are hypotheses, it may interfere with pathogen growth. There are certain proteins, like heat shock proteins, that will be activated at different temperatures. So if we have an increased temperature, it'll activate these heat shock proteins. Then these heat shock proteins can then go on to do something else that may be involved in the healing process. Interferes with pathogen growth may slow them down. Different proteins, so different proteins may work differently, either better or worse. But here's the important thing. This is the important thing to remember. Without knowing why we get a fever specifically, we know that when there is a fever, that indicates a disease state and possible need for medical treatment. So as healthcare providers, we want to make sure that if there is a fever, or that a fever is increasing, that's kind of an indication that something may be getting worse now. Now, sometimes fevers, kind of, oscillate, but in general, that's something we really want to monitor for that reason, because it indicates a disease state. So manifestations: anorexia, so loss of appetite; fatigue, tiredness, malaise; not eating and resting conserves energy the body needs to fight off invaders. So we can kind of say, okay, well, if you don't eat, then you're conserving energy. So that's one thought. Now, chills. Okay, so chills, I'm pausing here because chills are important. So people feel chills. Okay, chills are associated with a fever. Okay, and why? The question is why, and this is really important to understand. So when I was a kid, I thought that, oh, I'm getting chilly, I feel cold even though I have a fever. Well, that doesn't make any sense. It's, oh, okay, it's because my body's so hot that the normal air feels colder to me. That's not it. That's not what chills are. Chills are just like if you go outside on a winter day and it's really cold outside, you feel a chill. You start to shiver. Why? Because your body is trying to increase the temperature. Okay? If you're trying to increase the temperature, that means you're trying to warm itself. Okay? So that's why someone who is developing a fever has chills, because their body, their hypothalamus, okay, in their brain, their hypothalamus has changed the set point. It's saying, oh, nope, we're not going to be at 98.6 or 37 degrees anymore. Now we're going to increase the temperature. We want it to be higher. We want it to be 38, 39 degrees now. And so it sends out that typical response of what you do. What do you do when you're cold? Well, you get chills. You start to shiver. Okay? Your teeth start to chatter, things like that. And so you have a normal response to try to increase the temperature. What this means, and the important thing about what this means, is that if someone is experiencing chills, that means that their temperature is actively increasing at that point. Okay, so if someone is experiencing chills, then you're going to want to pause and you're going to want to say, okay, we need to start taking vital signs again. We need to start taking your temperature to see where this is going, to see what's going to happen here. Now, once you've reached your new set point, the chills will fade. Okay, so you really feel the chills during this phase where your body is trying to increase the temperature. And once you've reached that new set point of 39 degrees, well, the chills, you won't have the chills as much. Now you still want to maintain it, so you might feel it a little bit, but the severity will definitely go down. So chill from the body's attempt to warm itself. It triggers shivering, and it's when the body reaches a new set point, or when the body reaches a new set point, the chills stop. Okay, so what causes this? Pyrogens. There are chemicals called pyrogens that are released from macrophages or endothelial cells that will say, hey, okay, we're going to raise the body temperature. Okay, prostaglandins formed in the hypothalamus will change the set point. So this is another reason, prostaglandins is another reason why aspirin NSAIDs can reduce fever, because they block the production of the prostaglandins, and that will cause the fever to drop. So see how all of this stuff is working together, and you really need to understand these things because you can form a whole bunch of test questions just based on those concepts. You know, if somebody's feeling chills, what's going on, that kind of thing. Somebody's taking an aspirin, why is that reducing fever? All of those things. So fever mechanism caused by release of pyrogens. Fever triggering molecules may be released from host macrophages, pathogens, endotoxins, or endothelial cells. So it can be released from a whole bunch of different places. But the temperature control usually takes place in the hypothalamus. I mentioned this earlier. Remember it. Okay, it's a different set point. That is what's going on. The hypothalamus now has a new set point, and so it's trying to get the temperature up to, in this case it says 41 degrees Celsius or 105 Fahrenheit. That's the max generally, although it can go higher than that. But that's what's going on. So release of cytokines or endotoxins triggers prostaglandins. There's that word prostaglandin again. Prostaglandin release in the hypothalamus to form a new set point. And then we just have the normal, what happens when we're cold. You know, our body temperature is below what we want it to be. So we go through this physiological so cell metabolism goes faster to produce, you know, more ATP, burn more glucose, and get the temperature up. And behavioral, shivering, covering up, need a blanket. Your body's trying to get itself hotter. Responses continue until the new set point is achieved. Now the problem is neurological damage can alter the body's ability to maintain a constant temperature. So sometimes we see that in head injuries, things like that. So in general, it's okay maybe to control a fever or a temperature, a fever, yeah, because it can cause damage, and it kind of makes us feel crappy. So if we already know what's going on and some kind of treatment is, you know, imminent, taking place, then controlling the fever is in general a good thing. Okay, and then I mentioned earlier that sometimes fevers behave differently. We're not going to get into this in any detail, but you can see that there are intermittent fever, remittent fever, sustained fever, or relapsing, where it goes down and then it goes back up again. And these can be associated with different treatments, but we're not going to go into that detail. We are instead going to end this section.